Okay, Year 9, let's talk about this Year 9 exam and go through the questions here. The first part of question 1 is about recalling facts about cells. You've got 3 parts of cells and we simply need to match them up to their job. So if you've got this wrong, you must memorize these facts. The nucleus's job is to control the activities of the cell. The mitochondria's job is to release energy for the cell, and that energy is released through the process of respiration. And cell membrane's job is to control which substances can pass in and out of the cell. Part B gives you a diagram of a plant cell. I can see that it's a plant cell because it's got the features that I've learnt. So I know that plant cells have cell walls on the outside and have chloroplasts for photosynthesis. I'm being asked to pick out those 2 bits in the question here because it says which 2 parts are not found in animal cells. Now, an animal cell does have a nucleus and does have a membrane and does have cytoplasm, so there's only 2 correct answers from this diagram, which are cell wall and chloroplast. An alternative would be to spot that this cell has a vacuole, and even though it's not labelled, a vacuole is an acceptable answer to something which is not normally found in an animal cell. Okay folks, quite tricky question 2, tripped quite a few of you up, and we're just looking at all the information here which is describing how to do the osmosis required practical about finding out the effect of changing the concentration of sugar solution on either the size or in this case the mass of potato tissue. When we get questions like this, I think it's quite often worth skipping straight down to the question you're being asked and then looking for the information you need. So the question says calculate the value of x in the table above. I'm looking to calculate this number here. I'm seeing that it is the percentage change, and the percentage change calculation would be how much does the mass change by compared to where it was at the start. So the maths I need to do to answer this question is the change in mass, which is 0.15, divided by how much its mass was at the start, and then you'll recall from your maths lessons we multiply that by 100 to turn it into a percentage. The correct answer is 11.1%. Okay, on to part B, and you can see we're being asked here why would we calculate the percentage change in mass as well as the change in grams. Now the answer here is very specific, and it's because no matter how hard you try in this practical, there's no way you can make all of your pieces of potato identical masses at the start. You can see they're quite close to each other. So in order to make it a valid comparison between the different pieces of potato, rather than saying how much has the mass changed by, we're saying what percentage change has happened compared to its original slightly varying mass. So a good answer here would be, because they have different masses at the start, so because the masses were different at the start, by calculating the percentage change, it allows us to make a direct comparison to say what effect has changing the concentration of sugar solution had on the mass of the pieces of potato that we've left soaking in these tubes full of sugary water. Okay, Year 9, slightly tricky graph here. We've got negative and positive numbers. Now, the most important thing, and quite a few of you made the mistake here, was to plot the incorrect column on the x-axis. We're looking at the percentage change in mass, and this scale has been put here for you already. However, the x-axis needs to be filled in. Now, we put the independent variable on the x-axis, which is always this column in a results table. So whenever you see a results table, this is the values that go on the x-axis, and the thing that we are investigating and measuring is going to go on the y. So I need to put the concentration of sugar solution in an even scale across my graph, or on the bottom here would also be fine, and I need to label what it is. So I'm looking at these numbers. I need to go from 0 to 0.8, pick a nice easy scale. One of these big squares should do us for 0.2 as we go across here. Okay, so I'm going to write the numbers down the bottom so that you can see them more easily. 0.2, that means 0 is here, 0.4, 0.6, 0.8. It doesn't matter that we haven't gone all the way across. So you can see that I have labelled that axis as matching the label that's on the top of the column, the concentration of sugar solution in moles. So this graph is going to say to me, how does the concentration of the sugar solution affect the percentage change in mass, which is the point of the experiment. Okay, so when we're plotting graphs for GCSE, the points that we put on the graph need to be accurate to within half a small square. So we need a sharp pencil, and we need to be plotting as accurately as we can. I'm looking at my table at 0 concentration. The percentage change is 16.2. I need to work out where that goes. So I know it's above 15, but how far above is it? I'm going to count the squares. There's 10. So every 2 squares is 1. 16, 17, 18, 19, 20. Once I've checked the scale, I'm going to go to 16, and it's 16.2, so it's just above, and I'm going to put a cross on my graph. I'm going to repeat this process for the other points. Now this point here, I calculated, and I've got 11.1%. If you made a mistake in the calculation, you won't have the correct number here, but that won't matter when you come to have your graph marked. Okay, so you can see that I've put my points on the graph now. I'm looking back in the question, have I chosen a suitable scale? Yes. Have I labelled my x-axis? Yes. Have I plotted the percentage change in mass? I have. And I need to draw a line of best fit. Now there's 2 options here. The line of best fit could be a smooth curve, or it could be a straight line. I'm looking at my points and thinking my best fit here is going to be a straight line. I'm imagining a line which goes as close to as many of the points as possible, and I'm going to draw that line onto my graph. Okay, part D is relating to our graph that we've drawn. It's asking us to estimate the concentration of the solution inside the potato cells. Now the bit of science we need to understand here is that if the sugar solution outside the potato in the test tube is equal to the concentration of the cells that the potato is made out of, that there'll be no overall movement of water by osmosis. We'd say that the 2 concentrations are the same, and the word for that is isotonic. So the isotonic point on my graph is where my line of best fit touches the 0 change in mass line. So it's this point here. And if I relate that to my scale, the concentration would be 0.5 moles per decimeter cubed. So if you've got an answer between 0.45 and 0.55, because everybody's lines of best fit will be slightly different, you will be within tolerance and get that mark. Okay, we often get questions like this where we're being asked to suggest errors, and errors happen in experiments usually because we haven't measured something very well, or that something is happening that we can't control. So when we're thinking about here, I would suggest the things where you've made measurements, for example, making up the different concentrations of the sugar solution, how much you dry the pieces of potato before you remeasure their mass. Both of those things could go wrong. And then within the method itself, you might have problems with your balance. Is it set to 0? Is it accurate enough to measure the change in mass? And possibly, is the experiment set up long enough so that water actually evaporates from the tubes and changes the concentration of the sugar? So I'm going to go for the 2 measurements because that's a good rule. Errors are caused when you make mistakes with measuring. I'm going to say that the concentration of the solutions were not made up correctly, so they're not what we think they are. And I'm going to say that when we took the potatoes out, they weren't all dried equally, and so there's variation caused by how much we patted them dry with the paper towels. Okay, so you can see my answer is that the concentration of the sugar solutions were not accurately produced, and that the chips were not dried by the same amount each time before the mass was remeasured. Okay, the final question in the biology section of the test. We've got a image showing part of a root from a cress plant. It looks like this image is at a very detailed and very high magnification, and so the type of microscope must be an electron microscope. Remember the facts that the magnification and the resolution of electron microscopes allow us to take more detailed and more magnified images. Okay, so this question about a magnification calculation, all right, I'd be thinking to myself, I'm going to need my equation for microscopes here, and remembering that the image size is related to the actual or the real size and the magnification. Image size, actual or real size, and magnification. The question is asking us to work out the real length of the root hair, so that's this. And so I need to know the image size and the magnification to work this out. I need to measure from the picture the length I'm being asked to work the magnification out from. And I can see on my ruler that this is 3 centimeters, which is 30 millimeters long. You'll see why I put it into millimeters in a minute. I'm looking at this the question and seeing that the magnification is times 200. And I've got this information here that there are 1000 micrometers in 1 millimeter. I've got to calculate the real length of the root hair, so I need to do how big is the image divided by the amount of magnification. I'm being asked to give my answer in micrometers, so I need my image size to also be in micrometers. So I know that my image was 30 millimeters, but that is going to cause me an error unless I change it into micrometers using this fact here. 30 millimeters is 30,000 micrometers. And I'm going to divide that by the magnification, which it tells me in the question is 200. When I use my calculator to work that out, it gives me a real length of this root structure of 150 micrometers. Okay, so have a look at what you've done. Did you not convert into micrometers? Did you get your numbers the wrong way round? What are you going to do next time to make sure you don't make the same mistake if you made one? Okay, folks, final bit of our biology questions here. We're being given some information. Root hair cells take up water from the soil, and this is an explain question. The command word says explain one way in which the root hair cell is adapted to this function, the function which is to take up water from the soil. So we shouldn't use this take up water from the soil in our answer, but we should think water osmosis, movement of water into the root is going to be by the process of osmosis. I'm going to get that into my answer. And I'm thinking, what is special about root hair cells? They are this shape that we know, with these long bits that stick out into the soil. Okay, it means there's a larger surface area of cell membrane. So what is the adaptation? The adaptation is that these cells have a large surface area. What is it for? So you can do more osmosis and take in more water.